Good afternoon and welcome. In this presentation, I will summarize our HIMSS 2026 paper on a subject-wise wavelet scattering transform and AlexNet framework for EEG-based Parkinson's disease classification and candidate channel screening. The motivation is straightforward. Earlier image-level benchmarks showed that the WST-AlexNet representation separates Parkinson's disease from healthy controls very effectively. However, health informatics workflows are ultimately interpreted at the level of the participant, not the individual image. In this work, we therefore re-evaluate the benchmark selected channels using leave one subject out validation, and we then screen all available channels to identify candidate electrodes for future EEG Parkinson's pipelines. The talk is organized into 4 parts. First, the motivation, why participant-level assessment matters for EEG-based Parkinson's disease classification. Second, the framework, how we combine wavelet scattering transform images, AlexNet, and a subject-wise leave-one subject-out design. Third, the results, the targeted reassessment of the benchmark channels, followed by the exploratory alt-channel screening. And fourth, the value, the candidate channels we identify and the contribution to health informatics. The presentation is designed to run automatically for remote playback and to leave time for questions afterwards. Let me begin with why this problem matters. Parkinson's disease affects motor, cognitive, and quality of life outcomes, and there is strong interest in accessible markers to support assessment. Electroencephalography is well suited to this role. It is non-invasive, portable, comparatively inexpensive, and it offers high temporal resolution. The key point for model assessment is the unit of evaluation. In a clinical or screening context, the decision is made about a participant, not about a single image segment. When many images or segments are generated from one participant, the evaluation protocol must make clear whether the reported performance reflects image-level pattern separation or genuine subject-level assessment. Our goal in this study is a subject-wise EEG Parkinson's assessment workflow that respects that distinction. There is a further, technical reason this matters. If segments from the same participant appear in both the training and the test set, the model can learn participant-specific characteristics rather than disease-related patterns, and the reported accuracy can be optimistic. A subject-wise protocol removes that leakage and gives a more honest estimate of how the model would behave on a genuinely new person. Our starting point is the previous WST-AlexNet benchmark. That study reported strong image and segment-level performance, 95.79% accuracy on the Iowa dataset and 99.84% accuracy on the San Diego dataset. It also identified a FZ and a F4 as informative channels. We do not dismiss that evidence. Those results demonstrate the representational strength of the approach. Instead, the current work extends it by asking a more demanding participant-level question. How do the benchmark selected channels, and indeed all available channels, behave when each subject in turn is held out as the test unit? In other words, we convert a strong image-level benchmark into a subject-wise analysis. It is worth stating plainly that subject-wise accuracy is expected to be lower than image-level accuracy because the held-out participant is entirely unseen during training. The value of the exercise is therefore not a higher headline number, but a more faithful measure of participant-level generalization. The paper makes 3 contributions. First, as subject-wise reassessment, the benchmark selected are FZ and F4 channels are evaluated using leave-want subject-out folds so that performance is reported at the participant level. Second, an all-channel screening, 60 Iowa channels and 32 UCSD channels per condition are ranked by subject-level accuracy under the same framework. Third, candidate channel discovery are FZ, OZ, and F8 emerge as the strongest observed candidates across the 3 classification tasks. Taken together, these 3 elements form a compact and reproducible health informatics pipeline for EEG Parkinson's model assessment and channel screening. The study draws on 2 EEG Parkinson's datasets, giving 3 subject-wise classification tasks. The Iowa dataset provides a healthy control versus Parkinson's comparison, with 14 Parkinson's participants and 14 healthy controls recorded across 6 to 4 channels at 500 Hz under eyes-open conditions, yielding 28 leave-want subject-out folds. The San Diego, or UCSD, dataset contains 15 Parkinson's participants and 16 healthy controls recorded across 32 channels at 512 Hz, and it includes both an off-medication and an on-medication condition, each giving 31 folds. Across the all-channel screening, 126,324 usable wavelet scattering transform images were available, spanning 124 comparison channel groups. Both datasets are approximately class-balanced, which is helpful because it lets us interpret accuracy directly. Segmenting the continuous recordings into short windows is what produces the large image counts used for training, while the leave-want subject-out structure ensures that those images are always partitioned strictly by participant. The method comprises 6 linked stages. First, each EEG channel signal is segmented into short time series. Second, the segments are represented as wavelet scattering transform or time frequency images. The scattering transform applies cascaded wavelet filtering, a modulus nonlinearity, and low-pass averaging, which together produce stable, multi-scale image features. Third, AlexNet-based processing extracts features from these image representations. Fourth, the leave-want subject-out folds are constructed so that every image from a held-out participant is assigned to the test fold and none of the participant's data appears in training. Fifth, the segment-level probabilities from the held-out subject are averaged to form a single participant-level prediction. Finally, every common channel is evaluated under this subject-wise design and ranked by accuracy. A practical advantage of the scattering transform is that it is stable to small deformations and time shifts, which is desirable for non-stationary EEG. And because the representation is fixed rather than learned, it reduces the number of parameters that must be estimated, which is helpful given the modest number of participants available. We run 2 complementary analyses under this design. The first is a targeted leave-want subject-out reassessment of the channels reported as informative in the earlier benchmarks, FZ for Iowa and F4 for both UCSD conditions. The second is an exploratory all-channel screening, which evaluates every common channel, 60 channels for Iowa and 32 channels for each UCSD condition. In both analyses, the fold structure is identical and strictly subject-wise. One participant is held out, and every image belonging to that participant forms the test unit. This is what makes the reported figures participant-level results, rather than image-only performance summaries. This paired design is deliberate. By holding the representation, the network, and the fold structure constant, any difference we observe between analyses can be attributed to the choice of channel, rather than to a change in method. These are the targeted leave one subject out results for the benchmark selected channels. For Iowa, channel FZ reached 67.86% subject level accuracy. For the UCSD off medication condition, channel F4 reached 58.06%. And for the UCSD on medication condition, channel F4 reached 48.39%. These three values provide the participant-level reference point for the channels emphasized in the previous benchmark. On the chart, the blue bars show these targeted benchmark channels, while the orange bars preview the best exploratory channel for each task, which I will explain on the next slide. As one would expect under a strict subject-wise protocol, these figures are more modest than image-level benchmarks. The on-medication condition is the most challenging of the three tasks, which is consistent with medication reducing the electrophysiological differences between the groups. The exploratory all-channel screening applies the same subject-wise framework to every channel and ranks them by subject-level accuracy. For Iowa, FZ remains the strongest candidate at 75.00%. For the UCSD off-medication condition, OZ becomes the strongest candidate at 74.19%. And for the UCSD on medication condition, F8 becomes the strongest candidate at 70.97%. The ranked lists on this slide show an important pattern. The leading candidate channels differ across dataset and medication condition. That variability is precisely why the all-channel screening stage adds information that a fixed pre-selected channel set would miss. I would ask you to read these as observed exploratory rankings on modest cohorts rather than as definitive claims about the underlying neurophysiology. That said, the recurrence of frontal and occipital sites across tasks is encouraging, and it motivates focused confirmation in larger samples. The confusion matrices make the subject-level decisions explicit. Here, healthy control is the negative class and Parkinson's is the positive class. Rows are true labels and columns are predicted labels. In the targeted analyses, Iowa at FZ gave 11 true negatives, 3 false positives, 6 false negatives, and 8 true positives. UCSD off medication at F4 gave 9, 7, 6, and 9. UCSD on medication at F4 gave 8, 8, 8, and 7. In the exploratory analyses, Iowa at FZ gave 13, 1, 6, and 8. UCSD off medication OZ gave 11, 5, 3, and 12. And UCSD on medication F8 gave 12, 4, 5, and 10. The key observation is that the exploratory UCSD channels improve the balance of correct subject-level decisions relative to the targeted F4 channel. Reading the matrices in this way is useful because two channels can share a similar overall accuracy and yet differ in how they trade false positives against false negatives, which is an important consideration for any screening application. This slide places the results in their proper context using the same representation family but a different evaluation unit. The prior image and segment level benchmark reported 95.79% on Iowa and 99.84% on San Diego. Its purpose was to demonstrate representation learning strength and channel context. The current targeted leave once subject out analysis reports 67.86% for Iowa at FZ, 58.06% for UCSD off medication at F4, and 48.39% for UCSD on medication at F4. Its purpose is a subject-wise reassessment. And the current exploratory analysis reports 75.00% for Iowa at FZ, 74.19% for UCSD off medication OZ, and 70.97% for UCSD on medication F8. Its purpose is candidate channel screening. The key message is that protocol context matters. Image level and subject level figures answer different questions, and the subject-wise screening adds participant level evidence. Presented side by side, the three settings are not competitors. Each answers a distinct question, and reporting them together is precisely what allows a reader to judge the framework fairly. From a health informatics perspective, the practical value lies in how the framework is organized. It brings together, in a single reproducible workflow, 4 elements: wavelet scattering, transform image construction, AlexNet-based image processing, subject-wise leave once subject-out assessment, and accuracy-focused channel ranking. It also links the prior representation learning benchmark with participant-level validation. In terms of candidate channels, FZ remained the strongest Iowa channel, while OZ and F8 emerged as stronger UCSD candidates for the off-medication and on-medication comparisons respectively. The framework provides a direct comparison of targeted and screened channels and yields candidate channels that can be taken forward to larger cohort evaluation and multi-channel extensions. I want to be careful not to overstate the clinical readiness of these results. The cohorts are small, and external validation on independent data remains essential. What the framework offers today is a disciplined and reproducible way to compare channels and protocols on an equal footing. To conclude, this paper presents a subject-wise WST-AlexNet framework for EEG-based Parkinson's disease classification and candidate channel screening. The targeted leave once subject out analysis re-evaluates the benchmark selected at FZ and at F4 channels, while the all-channel screening identifies at FZ, OZ, and F8 as the strongest candidates across the Iowa and UCSD tasks. More broadly, subject-wise evaluation shifts the level of evidence from image-level patterns to participant-level model assessment. Thank you very much for listening. I would be glad to take any questions and to discuss the work further. We hope that the subject-wise protocol and the candidate channels reported here provide a useful reference point for future EEG Parkinson studies.